Good morning. I think we'll get started here. Welcome back to day 2 of the Rare Patient Advocacy Summit. Thank you for joining us in Track 3, Patients as Partners and Drivers, sponsored by Shire. I'm Michael DeLeon, and I will be your track host for this morning's session. I'd like to also welcome those of you joining us remotely through the live stream, and would like to remind everyone joining us to download our mobile app in order to use some of our highlighted features like the live Q&A. Today we'll be joined by Hans Schlecht, attending physician of internal medicine and infectious disease at Baystate Health, Michelle Davis, executive director at International Fibrodysplasia Ossificans Progressiva Association, and Douglas Macdonald, director of research operations and scientific alliances at CHDI Management, who will be diving into a deep discussion on collaboration in research and why it's so important and what makes a collaboration successful no matter how big or small. We can't do it alone. Structures and governance for research collaborations. Let's welcome them up. First, Hans.
Yes? Great. I just want to set a timer. Thank you so much for being here. Thank you to the conference organizers. What I'd like to do is to cover my own family story, and I've got a lot of information, so I'm going to have to speak quickly. But my own background, our journey since March 2017, and then emphasize some of the lessons that I feel that we've learned, and the emphasis there is going to be on communication. Approaches to finding the cutting edge, and then review some examples from our own experience that maybe illustrate the points I'm trying to make. So I have a medical degree, so I've got a foundation to sort of cope with the rare disease in my family, internal medicine, infectious diseases training, and in fact did a 2-year program in clinical investigation during fellowship. I've had NIH funding and collaborated on Gates Foundation funding, and I actively think about research. And in March of 2017, our whole exome report gave us an explanation as to why my little guy was developmentally delayed and fell into the category of non-syndromic intellectual disability. And this was a SYNGAP nonsense mutation, SYNGAP being a very critical protein in neurodevelopment. And these children, adults, are affected with intellectual disability, usually from moderate to severe, autism features, epilepsy, behavioral difficulties, sleep disturbances. So at least we had an answer, right? We had an explanation. And what I hope to emphasize for the lesson is that you've got a topic that you are a personal expert in. So you're going to tell your story and be able to provide it, share it, and let someone else take it with them. As much as you can, become an expert in your disease, the research, similar diseases, and similar research, and then who you're going to converse with. This is sort of, obvious in terms of the doctors you interact with, and here at conferences, the people and the advocates, and then where to do it, like we are today. So this is where my story begins, in the sense we were at a extreme parenting meetup, as it's referred to, and the Durrett family, who allowed me to share this with you, provided a 1-page handout. And I think it's a great lesson that they deserve full credit for doing, in the sense that the front side gave a story for their darling daughter, and on the back side was her whole exome sequencing, which is really the crux of important information. And obviously this is going to depend on what topic and disease area you're interested in. But here's a 1-page, and no one is going to say no when you, can I give you 1 page on my story, on my disease that I'm interested in? They'll take it with them. They'll look at it a second, a third time, and percolate. And the research ideas, maybe there's a eureka moment. Because for me, when they described their daughter, she was 10 years old and she was functioning at the level of a 6-year-old. And that's something I can only hope for or dream of for my little guy in a disease where it's moderate to severe intellectual disability. And you say to yourself, wow, that's really interesting. She is interesting. And then you look at her whole exome and you say, her SYNGAP mutation is a deletion right at the catalytic site. Wow. That's really interesting. And I spoke about it with someone else, and I'll circle back and give the follow-up on it. But this allows your story to travel, okay, and be returned to again and again. It must be daunting for a lay person, and one of the worst looks I've seen in a person's eye was the desperation in a parent. Science has been my emphasis educationally for a long time, for others not so, and you have to do the best you can to learn that disease. Get a good review article, do a deep dive on it, write it out in your home script, make analogies to your interests. Whatever it is, you've got to become an expert so that you can have that intelligent conversation, follow the conversation, understand the lingo. If you can get as far, learn the research that's going on. If you can get further, draw analogies to similar diseases. For us, that might be the rhasopathies or the infantile encephalopathies. Okay, so go as far as you can. There's numerous ways to do this and to find that information. Obviously, Google and setting scholar alerts. YouTube has great videos on the FDA, and PubMed as well. And PubMed is where, it's the library of scientific information out there. You punch in SYNGAP, boom, you come up with 1,000 articles. You can hit the drop-down box at the top for PubMed, and the next thing you know, you can lose, I would say hours, but it's days, weeks, and months because science has gone so deep. The gene, the amino acid sequence, the structure, the RNA-seq data, on and on and on. And it's really a testament to what the NIH has done for science, the US, the world, and it's truly amazing. Advocate voices matter, and the FDA made this clear. A year ago, I attended a rare diseases public workshop, and those materials are available online and are well worth the effort to go through, to familiarize yourself with the FDA and their format of approaching rare diseases and what they want from you and what they want from advocates and what advocates can do. Who is doing what so that you can talk to the best, okay? And you're going to find the authors in PubMed. You can look at clinicaltrials.gov and make connections and write down a hit list of the people who you want to talk to, whose ear you want to bend, who's doing the basic science, the clinical research. PubMed. I also want to bring up the BRIMR.org and NIH Reporter, and I'll get to those in the next slides. So the Blue Ridge Institute of Medical Research has collated NIH funding and broken it down by subject in a user-friendly, easily accessible kind of way. So you can look at the basic science departments, clinical science departments, click on that, and you'll see just how much is going where. So naturally, I'm going to show you neurology, and I say to myself, wow, UCSF is killing it. I mean, $56 million of the creme de la creme money is supporting their neurology. If I'm within a 2-hour drive, I get in the car and I go there, unless I'm closer to, Stanford and USC Davis, et cetera. So on the East Coast, it's Columbia. It pays to put in the effort to go to the bleeding edge and have them interested in your care, in your advancement. NIH Reporter, a public way that the US taxpayer, ostensibly that was the grounds, can see where the money was going. And if I punch in SYNGAP right here at the text search, I get my list of the experts. Dr. Koba, who spoke yesterday, Dr. Hugeners, Dr. Rumbaugh. These are the leaders in the field. And now I can click on their project and say to myself, all right, so-and-so is looking here, 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 and I'm getting a background, and I'm peeking over their shoulder in a way at the grant that they're working on. Top right, I click on the name, that's his email address. So now I've got in my sights the person I want to reach out to. And this is obvious, I'm not going to emphasize it too far, but parenting meetups, the FDA, keep talking. What I will emphasize is be open-minded, but you have to also trust but verify. You will hear some whoppers come out of people's mouths, and everybody's human and trying to find answers and reach potentially for an answer. So trust but verify. Keep a Rolodex of the earnest. If I have an idea, I reach for Dr. Koba, Mike, Aaron, whoever I know is informed, has an open mind, give me honest feedback without some swaying in either direction. And keep in touch with the people in your field that are earnest, that are in it with skin in the game. Look at what others have done. You do not need to reinvent the wheel. So for me, I thought a great analogy was Leslie Gordon and the Progeria Research Foundation, in the sense that, clear as day on their website, they broke it up into a registry, a cell and tissue bank, medical and research database, as well as a diagnostics testing program. And that's an outline to use. And now I'm going to give you examples over the past, 18 months that hopefully illustrate ideas and making connections. So you get your whole exome result. For me, a nonsense mutation, obviously nothing else is on my mind. Deep dive on Google and follow every lead, and you come across adalurin, right? And this is a drug familiar to the Duchenne's and the cystic fibrosis field as a nonsense mutation read-through agent. The idea that the stop sign in the gene, you're just going to blow right through using this medication. So within days, I'm emailing, I would like to start a conversation about getting expanded access to adalurin. And obviously the answer is no, you're the dad. No, it doesn't matter that you're a doctor, he's got a medical team. The answer is no. Okay, fine. You wait for your medical appointments, you get in with the expert. Are you willing to reach out? Yes. Okay, use some back channels, try to get some answers. The answer is still no, but you know what? We're looking at early 2018. Great, the family, we move up to Massachusetts, put in the effort, drive to Boston Children's, meet with Dr. Pridori, our new pediatric epileptologist, genetic epileptologist. Hey, would you mind applying for expanded access? I have a letter here. Sure, that makes sense. Let's give it a try. They actually, PTC Therapeutics said yes. And we had already braced ourselves for another no and would have then used the FDA's personal importation license program to get the drug ourselves at tremendous expense and a lot of sleepless nights probably. But thankfully they said yes. Next thing you know, it's at the FDA, turning it around within 2 weeks. Nothing but applause there. And then it goes to the Boston Children's IRB. We sign the consent forms, and 3 days after his 3rd birthday in March, he starts the drug. And so what I'm hoping to do is show you this video because it'll let me get a drink of water, and also it will stimulate a point I want to make.
Yes. Swing.
Ooh, look at you go!
Swing. More. Yay!
So that's my nonverbal kid on Friday when it was emailed to me expressing verbally his wish. Now, what I am stuck saying is, I don't know why. There are 4 explanations for the results of a drug trial. There's fraud, bias, chance, and drug. And unfortunately, I don't know if this is chance or drug. And the point I want to make is, if you have a registry, if you have a natural history study, you've got to get in it. It is so important. I have no sense of what the trajectory is for development for a SYNGAP kid with a nonsense mutation in exon 9. I have no idea, and that's asking a lot. I shouldn't say we, the connection was made between human disease and SYNGAP mutation in 2009 in a New England Journal paper. It's only been 9 years, and that may sound, wow, 9 years is long, but science takes time. But if you have the opportunity to participate in a registry or a natural history study, you've got to do it, because it helps answer the question is what to expect. And is this normal development? Is this facilitated development? I just don't know. Another example of the translational research I alluded to with the Durrett family. So when I sat down with Dr. Kobai, emailed him, found him an NIH reporter. He was gracious enough to meet with me when he was visiting the Broad Institute. And over an hour-and-a-half conversation, he made the offer of, well, if I can get a sample of your son's blood, I can make iPSCs and look at his genetic mutation in my lab. Great, you bet. IRB through Boston Children's, sample taken, shipped to USC, stem cells being looked at. Then when I meet the Durrett family and Peyton, who's functioning at the level of a 6-year-old, it clearly needs to be studied. It clearly is a natural experiment that needs an explanation. How do you think, Dr. Koba? I really want to look at that. Next thing you know, Peyton's got a sample sent to USC, and he's looking at that as well. And if it leads to something, wow, that's just great. These connections really can lead to something. So we've got to think, be in touch, email me, you've got an idea, great. A year ago, I sat in a talk by J. Lee Nelson, who's an expert on microchimerism. And I said to myself, wow, you know, I had met David Feigenbaum through Grand Rounds, invitations, and talks and so forth. Dave, what do you think of this? Here's a YouTube link to her expertise and her lecture. Wow, that's intriguing. Who knows where it leads to? You just don't know, but an email is so simple that just provide that and people can run with it. So the other idea that I'm trying to emphasize and talk about is that SYNGAP is present within the plasma. This was a survey when they were doing it 10, 11 years ago of plasma proteome. What are the ingredients within plasma? And sure enough, SYNGAP is there, 3 citations, clear as day. But who's going to go looking? Is this a biomarker? Does it reflect what's going on in the brain? And it's a part of the conversation that someone hopefully will answer for me. I want to share this as an email in the sense that it shows you, maybe as an example, how to reach out. And so this is me reaching out via email to an Alzheimer's plasma biomarker researcher. And what I've done is I've drawn a connection. I've said, I've thought about this to a degree, but I have 2 short questions for you. You do not, and I don't want to be a black hole of need, okay? This person is busy. If you can answer this, how does it get into the blood, and do you think anybody would be interested in looking at SYNGAP? He replies, good questions, neuronal exosomes being butted off, and Dr. Getzel at University of Kentucky, here's a paper to read up on, thanks so much. Hey, thank you. So there's a thread now, and maybe I can come back to it and say, hey, we've got some money if you want to look. But short, sweet, to the point, and thought through. Last but not least, just over the past few weeks, I've tried to share this idea, and I'll bring it up here as well. People want ICD-10 code so that they're in the category, they can look across claims data, they can organize. And you know what? For us, you hear it again and again and again. And it could be tied to genetic information as well. So what I've pitched to some friends are offering like a Q99 code for other chromosomal abnormalities and pairing it to the uniprot protein label that's unique to that protein. So for example, with a SYNGAP mutation, someone, the medical geneticist, would upload a Q99, Q96 PV0. So you see that would be specific to SYNGAP. And the person, Dr. or Mr. Rosen, who was speaking yesterday on KIF1A, it could go in as a Q99, Q12756. And that at least would give the genetic mutation its own ICD-10 code. Good idea, bad idea, what do you think? Do I get help? I've shared it with my earnest sort of, sounding board and a global genes person. But, with rare diseases numbering 30 million people, we're bigger than the CDC and we can get our way. So I'm going to wrap up, and I'm sorry if I've gone over. I have a lot of people to thank, and essentially it's anyone who's ever had a conversation with me, and I hope you guys have luck with your conversations. Thank you so much.
Well, thank you so much for having me. My name is Michelle Davis. I'm the Executive Director of the Fibrodysplasia Ossificans Progressiva Association. You may hear it referred to as FOP. It is an ultra-rare musculoskeletal disease. We have about 800 to 1,100 diagnosed patients in the world, and we believe that the incidence is about 1 in 1.4 million. So we have a lot of patients to go identify. So we have been operating for 30 years. Actually, this is our 30th anniversary. Our organization was started back in 1988 by a woman who went to the NIH because she had this disease that no one really knew anything about. And when she was there, they told her, do you know there are other people that have the same disease as you? And she said, I don't. And they said, and this was before HIPAA, would you like their names? And so they gave her a list of 17 people and their contact information. She went home and she sat at her typewriter on her desk and typed each of them a letter. And 10 of them wrote her back. And it was when you had to pay for long distance, so they wrote each other. And they started this pen pal organization. And the next year, a dad of a little boy with FOP said, I think I might have a golf tournament. And he worked in finance in Boston, and he had a golf tournament that raised $25,000. And that funded the very first FOP research symposium in 1991. And so what I'm going to talk about today is that, our vision has never changed. Our vision is a cure for FOP, but we exist now kind of encompassing all of the things. So when they started the IFOPA, their goal was to connect with one another. And so we still do patient support and education and connectivity. We still raise awareness. We've got to find those other patients that we don't know who they are yet, that haven't been diagnosed. But our primary mission has always been, in getting to that vision of a cure, is focused on funding research. And in the beginning, it was literally just funding research. But what I'm going to share with you today is how that has evolved for our organization and the variety of things that we've done in the research arena to really help further research. So this is where we are today. We started by funding that very first FOP research symposium, and you'll see that up here in the upper corner that we are still hosting research meetings. Every other year, we hold a drug development forum where we invite academic researchers, pharma, we invite clinicians from the United States and all over the world to get together and to discuss FOP research, and it bounces between the United States and Europe every other year. We've been then in several years later, we gave our very first research grant to the University of Pennsylvania, which is now kind of where the father of FOP research is located, Dr. Fred Kaplan. And so we've been doing grant support of FOP research. Families raise funds, the IFOPA raises funds, and we support research primarily at the FOP lab at UPenn. We've had this long dedication there, and that continues. But we've also started expanding in the last couple of years. So a great deal of the work at UPenn has been discovery and basic science work, some translational research. But 3 years ago, we had a family that came to us and said, we just want to look for treatments. How can you help us do this? So now we have a program called the Accelerating Cures and Treatments, which stands for ACT for FOP grant program, and it only funds research applications that are focused on therapy development. And we've given over $500,000 to a variety of projects in the last 3 years. And then this year, we participated for the first time in the $1 Million Bike Ride. How many of you do that program? It's an incredible program with an incredible match. They do have a booth in the exhibit center, so if you have not stopped by there and learned about that program, it's incredible. And this year we raised enough funds and with the match to give away 2 new FOP research grants. And we actually held some scientific meetings, and topics that were addressed at that scientific meeting are now what we're funding through those grants, through the $1 Million Bike Ride. So like the circle is feeding itself, which is really cool. So as FOP research has continued, the gene for FOP was discovered in 2006. We are very blessed that there are many pharmaceutical companies and academic research institutions that are looking at various parts of FOP and how they can work to solve this puzzle. But what we've learned is that there are resources that they need that would accelerate research. And so we've decided in the last 2 years that the IFOPA is going to invest in those resources and develop open access research infrastructure pieces that they can use so that it's not proprietary information of any academic institution or pharmaceutical company. So we are literally just in the final stages of developing an FOP mouse model, and then we're also in the final stages of IRB approval for an FOP biobank, so that if any researcher anywhere in the world needs access to cells or tissue or blood or other biomaterials, that they can access it for shipping costs from the IFOPA. We also have a registry. So we have the only single registry for FOP patients in the world. It's very similar to the AT registry that they were talking about yesterday. One thing that's unique about our registry is we started with a patient portal. So that launched in 2015. In 2 years, we had 1/3 of the known diagnosed patients in the world in that registry. And it's a longitudinal study. So every 6 months, they are going in and updating and telling us us what has happened with their FOP in the last 6 months. We are in the process now, after seeking industry sponsorship, launching a medical portal. So now we'll have this very robust data coming from their healthcare providers that is adding to the patient's FOP story as well. There was a natural history study that was done by a pharmaceutical company, and through a partnership with a pharmaceutical company, they are also where there are shared patients depositing that data in our registry. So we've done a lot to make that, if it's going to be the only one in the world, we want to make it robust. It's translated right now in 7 languages. We're struggling with internet connectivity and how we get patients able to use that. So it's a question that's top of mind with us. So if that's one of your issues and you've solved it, we'd love to know how you're doing that. But I would say one of the things that we are most proud of at the IFOPA is the registry and how that's going to change research. And then the last thing that we're doing in the area of research is a prevalence project. So we are actually going to be investing dollars in counting the number of patients that we believe. Years ago, there was some work done to say that it was about 1 in 2 million. Some other researchers began to say, well, we think it's more prevalent than that, but we really don't know how prevalent. And so we're actually going to invest the resources in doing a prevalence study of our own. So that's kind of the complement of research work that we're doing. One thing I do want to mention, because I think it's very important, is you've heard me talking a lot about industry and pharmaceutical participation and support. And in 2016, we sat down and said, how should we, as a nonprofit organization, truly be interacting with pharmaceutical companies? And so we couldn't find guidelines. And so our research committee chair, Betsy Bogard, went through the task of actually writing guidelines on how we as a nonprofit organization should have a healthy, productive, but appropriate relationship with pharma. And so she's written those guidelines. They've been presented on here at the Global Genes meeting. And if you would like access to a copy of that, we're glad to share it with you. We're working on version 2.0, so it is a living document. As we're doing more patient advisory boards, we're seeing a need to really clearly define how those work and what our role is and what the role of pharma is, and so we're developing that as well. So I just wanted to show you quickly, as I wrap up, how we're structured. So this is our staff at the IFOPA. We do have a research director, an incredible gentleman, Adam Sherman, who spent 20 years in pharma, and he leads all of our research work. So the last slide that you saw, that is Adam's world. For several years, we kind of worked with Adam's role managing the registry, but as it started to grow and develop, and there were more requests coming in for data, and there were more requests coming as we started to launch the medical portal, we hired someone on a contract basis that actually runs our registry for us. As we need to, we employ statisticians to pull data and do analysis. We want to provide that information to the community as they need it, but we also want to protect the privacy of the patients. So we make sure that we fulfill any data requests that arrive. The other thing that I think that is important for you to know is that no one on our staff has a connection to FOP or has a family member living with FOP, that we rely heavily on our volunteer structure of the organization. And many of the volunteers in our organization are patients or they are family members that are helping guide our decisions. So we do have a research committee that reports to the board of directors. So I'll give you an example, like on our Act for FOP grant program, the scientific advisory board would actually review those grants. Those are medical experts in musculoskeletal diseases. They review and they rank blinded grant applications, and they select who the winners are, and then that goes to the research committee for approval, and then they send that to the board of directors to allocate the funding. So that's how we use our volunteers. We also have an International President's Council, which is made of people who run FOP organizations around the world. They're not official chapters of ours, but they are people that have either started an organization or who have said, I'm the organizer of FOP patients in Serbia. And we work with them because we are the only one that has paid staff of all these organizations. And so we say to them, if you're raising your money in your country for research, but you don't have the infrastructure we do to vet the research and to go through the process, then we'll partner with you, and whatever money you raise, you can be a part of this program and be a part of funding research. So they're excited when we're able to talk to them about what outcomes are happening from the research that they funded, and they're able to do that without having to do all the work. And then the last thing I'll mention is that we do have for our medical advisory board, for our registry, a medical advisory board. And that medical advisory board oversees the registry, they oversee the growth of the registry, they publish about the registry. When there are data requests that come in, then they also review those data requests and approve that. And they are also, the reason it's in red is because we're going to be expanding their role, and they're going to be taking on kind of a greater role as a medical advisory board for the organization in total. But that's a little bit about how we're structured. And then this is the last slide that's just kind of a high-level overview of, in the last 30 years, some of the key accomplishments. And you'll see that not everything happened at once. That's why I wanted you to see the slide. We were started in 1988. We started doing a medical symposium 3 years later. It took almost 7 years for our first research grant to go out. But you'll see, we had a role in identifying multi-generational families so we could identify the gene. But, our research committee wasn't established until 2012. So I just share this with you to say, if you are a small organization just getting organized, it might be overwhelming to see everything we're doing, but to know that it is a process and that I think the thing that I love about Global Genes and organizations like this is that we have the ability to connect with one another and say what worked and what didn't. And that's why I asked the question, are some of you solving the issue around, internet access and registry? So it's the beauty of being here, and if there's anything we can do to help you on your journey, we're glad to share what we've learned.
Just really quickly, before we hear from our last speaker, there's some open chairs down here if you guys want to come and find some seats. If everybody in the audience could raise your hand if there's an open chair next to you. You guys are welcome to come sit down.
Can I have the first slide? Thank you very much. Good morning, everyone. Thanks for coming and listening to us today. I've set my timer, so we're going to reserve the last 15 minutes of the session for questions and answers. I'm going to talk to you about how myself and a company called CHDI develops therapies for a rare disease called Huntington's disease. It's actually sort of known as the most prevalent rare disease. It's 1 in 10,000 people. But I'm going to tell you about that. I'm going to give you examples of how we interact with a number of different research organizations, whether it's biotech or big pharma or contract research organizations. And at the very end, I'm going to give you a little case study of how we enabled the initiation of a pivotal phase 3 trial that's going to start by the end of this year. So Huntington's disease is a monogenic disease. It's caused by a single gene mutation. The symptoms are, there are many. The hallmark are the movement disorders. It's also called Huntington's chorea to dance. But before you get those motoric dysfunctions, you have psychiatric and cognitive deficits as well. We heard a little bit about natural history trials by the previous speakers. This is a natural history trial called TRACK-HD. Dr. Sarah Tabrizi is at the University College London. She was the PI for it. And this is the same patient who came in for annual MRI scans. And if you really look right here, you're going to see the striatum shrinking and the ventricle enlarging. And that's the neurodegenerative process that goes on in Huntington's. And it's incredible that we have this data, because if we want to intervene and slow this progression, we now have this history, at least within this patient, and that study had about 500 or 600 participants, so there's a wealth of data there. CHDI is a not-for-profit biotech company. We're foundation funded. We're very fortunate. And we're solely dedicated to the discovery and development of therapies for Huntington's disease. I'm in our LA office, just north of here, but we also have an office in New York City and Princeton. However, all of these are office buildings. We work virtually. We are real, but we work virtually. It was a decision when we started about 13 years ago. It was going to be faster to buy than build. We didn't want to build our own labs. We didn't want to hire all the technicians that need to run the lab equipment. We were going to go out and find the best people to do what we thought needed to get done, and we were going to either pay them or enable them by other means to work on Huntington's disease. So I work with a bunch of colleagues. We're PhD or MD scientists. We're science directors. And we work with, as I said, biotech, big pharma, academic, but also something unique, contract research organizations. And I'm going to go through an example of each one of these and tell you how we work with them. But first, I should say we're about 100 people at CHDI. And we support up to maybe 500 people worldwide just to work on Huntington's disease. It's a very large effort. And this is a schematic of what we've done to create a portfolio of focus areas. So we're very interested in Huntington lowering. The gene that causes Huntington's produces a misfolded protein in neurons, and if we can stop the production of that mutant protein, we hope that we'll be able to slow the progression of the disease or even delay the onset. We're very interested in what that protein does, so the function. We know that there's neuronal dysfunction, so we study that and other targets, molecular targets. Regenerative medicine, stem cells. There's a new area called DNA handling and repair. Most of the time our neurons are really smart, and when there's a small mistake in our DNA, it can correct it. But what we found in Huntington's, that process isn't as, the fidelity of that process isn't 100%, so that can cause issues. And then we also look at systems biology, and that's really all the data that's out there. And we try to integrate that and come up with novel approaches to work on the disease. So the first example is working with an academic. So we've provided research funding to hundreds of academic PIs over the past 13, 14 years. Usually it's giving them money, giving them grants. And I shouldn't say grants. We actually give them contracts. This is sort of unique, and I think it's a model that you may want to adopt. They're on the hook for providing results. It's just not giving them money. Now, science isn't perfect. It may not work out, but I want a final report after a year. I want to see exactly what you did. If you made a cell line, guess what? That cell line is going to go to the Coriell Research Institute, and anyone working on Huntington's is going to have access to that. Because we paid for that as a not-for-profit foundation. I want that to be used by other PIs. I don't want them to have to make the cell line again. That just wastes time, right? We have these centralized biorepositories. We'll go out and get existing biosamples like antibodies, cell lines, or we'll make our own and deposit them there so they're free access. Patient samples, really important. So Huntington's is a CNS disorder, and so we have a big repository now of CSF samples. We've collected from about 200 individuals. We want to get at least 600 individuals to donate CSF so we can analyze it and understand the disease better. And of course, there's information. We provide different types of databases, but we also hold, and this is sort of a theme, no one else might have a therapeutics conference on Huntington's, but we do. So we gather academics, big pharma, and biotech together to discuss the latest and greatest and what's needed to go forward. Absolutely.
So when you talk about universities, some of the challenges that we face are the proprietary nature of the data. So you talked about
I'll repeat it.
as a nonprofit maintaining that
Right.
proprietary information. We're challenged with getting that through a university.
Right. So you're right. So when we do a contract with an academic institution, they own the data. I'm sorry, the question is, when you work with an academic, sometimes getting the data out of them isn't as easy because of data ownership. And it's true. You can have the university own the data, but use of the data can come back to the foundation in the form of a report and to be shared with other researchers. But ownership, publication rights, and things like that remain with the university. That's true. Yeah. Big Pharma, they can be your friend. They have worked for years and years and years on multiple targets and have small molecules, well, actually a variety of different modalities, but let's say it's a small molecule, a sharp tool to interrogate a particular pathway that could be involved in the pathogenesis of the disease you're interested in. So we approached Pfizer several years ago. They have something called a Pd10 inhibitor that's expressed in the striatum. That was that area, that piece of tissue in the brain that was shrinking. So that's an important enzyme there. And we thought by inhibiting it, it could actually help Huntington's. So we went to them and said, we know you have this compound. It was actually already used in a Parkinson's trial. And we said, would you give that to us? And we're going to spend the money and put it through our Huntington's disease models. And then in a year or two, we can come back to you and say, look, it worked. Or there's signs of improvement. Would you bring this to a clinical trial? And Pfizer said yes. They ran a phase 2 clinical trial called Amaryllis in patients on their own dime. We didn't have to pay anything else to get that into clinical trials. We did the preclinical work. We pitched this idea of using PDE10, and they went forward. Unfortunately, that trial didn't show efficacy, but the lesson's there. You can engage big pharma, identify their assets that could be leveraged for your disease that you're interested in, and just have that conversation with them. Another way that we work is, because we are research scientists ourselves, we work with these sorts of companies down here that are fee-for-service companies. So we have contracts with all of these. We say, hey, IRBM, we want you to run a high-throughput screen for this target because we're going to drive this forward. We're going to do what Big Pharma does. We're going to put medicinal chemistry on it. We're going to optimize the chemistry. So we've done that pretty successfully for numerous targets. And what we want to do with those, we're going to bring it to a certain level where Big Pharma might want to in-license those, either for Huntington's or maybe even another disease. And lastly, we work with biotechs. Biotech, they're very innovative. They usually have a very unique technology. And the case study I'm going to do next is based on our work with Ionis Pharmaceuticals. They are leading expert in antisense oligonucleotides. These things can bind the mRNA of Huntington and decrease the expression. So we're going to stop the production of the protein. We worked and supported a lab at UC San Diego, Dr. Don Cleveland's lab, and he published a great paper that showed sustained therapeutic reversal of Huntington's disease by transient repression of Huntington synthesis. That's a great title. Like, that's big. And what happened after that? Roche decided to form an alliance with Ionis to work on Huntington's disease. So all of a sudden we had a biotech company, we had a big pharma, because we supported this work at an academic lab who collaborated with Ionis to get that potential ASO therapy. Sorry. So this is the case study. And the case study is how collaborations can gate the progression to pivotal clinical trials. Ionis and Roche, as well as some other companies, were working on Huntington lowering. So this is a cartoon. The blue is the misfolded protein. We wanted to be able to measure that in CSF. Because if we're going to stop the progression of it, we need to know if we did in a patient. So we're going to take a CSF sample and we're going to measure the protein. The problem is the protein isn't very concentrated, and you needed special technologies to be able to measure it. So we worked with a number of different collaborators to basically run this test, okay? This is called a TR-FRET assay. It uses antibodies, the green and the red that bind the blue protein, and when they do that, it gives off this signal. And this is what had to come together to be able to do that. I had to get an antibody from Caltech up in Pasadena. I had to get an antibody from Novartis in Basel, Switzerland. I worked with IRBM, a biotech company outside of Rome. I worked at CHDI. And then I worked with physicians at University of British Columbia and University College London. And most importantly, we interacted with the families and the patients to provide the CSF samples that we wanted to investigate. Can we measure that protein? And we did. We published that whole international consortium. We published the ability to measure mutant Huntington protein in manifest patients versus pre-manifest patients. And obviously, you don't measure mutant Huntington in control patients, in controls, because they don't express the mutant Huntington protein. So we had this test, this assay, that could measure this biomarker to see if we lower Huntington in the CSF that is reflective of what's going on in the brain. Oh, sorry. So these are two neurologists. They are actually administering the Ionis antisense oligonucleotide therapy to patients. Dr. Blair Livet and Dr. Ed Wild, I should let you know who they are. And this data was presented at the Huntington Disease Society of America annual convention earlier this year. And what this shows is 2 different doses of the ASO and a decrease, as measured by that assay, of mutant Huntington protein in patient CSF. And what we think this means is that we're lowering mutant Huntington in the brains of Huntington's patients. And that's the cause of the disease. This is incredible. This is like better than we could ever imagine. And then this happened at the beginning of last month. Genentech, which is the American name for Roche, they just announced that they are starting 2 clinical studies to begin by the end of 2018. It's going to enroll 660 patients, and they are very serious about it, and they're going forward, and we're very hopeful that this may help patients. So this is my little, perfect timing. This is my conclusion slide. We can't do it alone. You saw what it took to make that assay, that test, and then to put it in the hands of Big Pharma so they could get that result and now invest. They're going to invest $200 million in that clinical trial because neurodegenerative trials are very long. But you can engage biotech, Big Pharma, CROs, and academic institutions. You can use research agreements, or you could just provide them disease domain knowledge and biosamples for them to enable their work. And I think it's very important to build multidisciplinary collaborations for hopeful success. So those are my slides, and I think we can come on up and we'll have a discussion. Thanks.
At this time, we're going to have Q&A. If there's any questions, if you could do me a favor and wait till I get the microphone to you before you ask your question. That way we can capture the question clearly for those that may be watching later on video. So anybody have any questions? Nobody has questions? There's one.
Hi, Nathan Peck from Cure VCP. We're a relatively new organization. My question's for Michelle. So you walk through kind of the life of the organization, and none of the staff have the disease or affected, when do you make that transition to bringing on what I'd call more professionals? It's kind of the entrepreneur dilemma. We're all patients in our organization right now, but when do you make that transition? When, is it a revenue threshold? Is it what is that point?
That's a great question. And when I started working for the foundation, for the association 2 years ago, it's one of the first questions that the patient community, when you start meeting them, ask. And so, for the IFOPA, it was the gene discovery in 2006 really catapulted research. And they started to see that there were many academic institutions, the number of them studying FOP was increasing, and that they were starting to get calls from pharma. And so that's when they decided that this is really something that even our research committee with some expertise can no longer really manage. And every day, myself and my team are like, how are we going to get everything done? The list just keeps growing because there's so much more engagement. So they went through a very specific strategic planning process before they decided to make the shift and hire professional staff. But it really catapulted when they started seeing more companies and academic institutions coming to them is really what the catalyst was for a real thorough strategic planning process and then making that decision.
There's one.
Hi, I have a question, I think for Michelle as well, about, you mentioned the pharmaceutical company that you were working with was able to share back the natural history data to your registry. We've run into at least one experience with a biotech company who really looks at natural history study data that they collect as a competitive advantage. And I was wondering what the impetus was, and was it a push from the families themselves, like, we are not going to participate in your natural history study unless you share it back? So
that's a great question. For us, we have the FOP Registry Medical Advisory Board that I mentioned on the slide that's circled in red. And that is a group of physicians from all around the world that advise us on the registry. And they were the ones that really put the pressure on the company to say, we want to make sure that if we're going to be encouraging patients to be in the registry, and then we are going to be using the medical portal, that the registry needs to be robust. And the way to make that robust is to take the patients we've been seeing in your natural history study and make sure the data is in the registry. So while patients did play a role in that, for us, it was really the Registry Medical Advisory Board that put the pressure on the company to do that. And I will say that our community research is very collaborative. And so, yes, are there moments where people are talking about intellectual property? It's happening at academic institutions and pharma. But for overall purposes, there's a real spirit of collaboration. But sometimes people say, so you're a nonprofit organization, how serious do you take all of this? I mean, we have lawyers that help us negotiate these contracts. Just like you said, we require that every grant we give out, there is, a full application. There's a 6-month, we pay out grants in 3 cycles. There's a 6-month report. If you don't turn in your report, you're not getting any more money. And there's a final report before you get your final payout. So yes, we're a nonprofit organization, but we take all of this as seriously as any business.
And so, right along those lines, actually, could you expand, because we provide some small grants and are looking to getting into big grants, and I guess the thing that we're struggling with is how much information do we get back from them, and it gets into the data rights and ownership. You're talking about reports and usage where the university owns it. It just seems to be a big gray area for us being a smaller org trying to do these things.
I don't know. Do you want to address that, or I mean, I can address it too.
Sure. So in our case, a lot of times when we give out a contract for specific work at an academic institution, it's been a conversation between us and them. And in a way, it's a joint research plan. So I think that's a big argument of, you owe us the final report, and we are part owners of that, and we're going to use that information to either fund the next person or to do whatever we need within our own institution. Yeah, I don't know if that totally, yeah.
So when you think about that research circle where I was talking about how different pieces are feeding different parts, when we give someone a research grant, we expect them to make a presentation at that drug development forum about what they've learned. Whether it was good or bad, we expect them to make some sort of presentation through a poster or through a presentation about what the results of that grant were.
I think there was another one here, yes.
Also the back row next.
Hi, I was just hoping you could speak to, there was a small bullet about HD and HD and the idea of the open access to the research that you all have.
Right, so again, it's a philosophy within CHDI that whatever we fund has to be shared with any investigator in the world. So HD and HD is a web portal where we're putting a lot of our RNA-seq data, and it's open access. And that's RNA-seq data on mouse models, so to enable preclinical research. It's more difficult to make patient data available through that portal because you need to sign certain things, but we do make patient data available through the different natural history trials that we've sponsored. But it's just a web portal that for preclinical scientists, that specific one. Yeah. I think in the back, do you?
For Michelle, I was curious about if you could share more about your International President's Council and how do you have guidelines, partnership, how you set that up?
Yeah, so the question was about our International President's Council. So it was started in 2007, so almost 20 years into the organization. Some families in the United States on our board said, we should form a process and a procedure and a place for us to have communications and a plan together with people that are running FOP organizations around the world. Sometimes you'll see in disease organizations that they'll set up chapters in the United States and then chapters in other countries, and that was never our intention. They are not legally bound to us. They are their own separate organizations. Some of them are registered in their countries in the UK or Canada or Australia or the Netherlands. Some of them are just starting that process, and some of them, like I mentioned, literally are a person, a patient or a parent who said, I will, if you have a message to tell patients about FOP in our country and you need someone to do that, I will do that for you. And so we 3 times a year, we do web conferences with them. So it's crazy. It's, people from, some people it's the middle of the night and they're on the web conference, and we all get on and, tackle whatever topic. So maybe we'll say, we're having a hard time getting, people from this country to do their follow-up survey. How can you help us do that? Or we'll brainstorm a question about a clinical trial that's coming. And then once a year, we get together in person because nothing really beats the value of that. And the IFOPA does pay for those representatives to travel to the United States or wherever that meeting is being held so we can spend a longer extended period of time learning from each other and finding new ways to support one another.
There was a question over here.
Hi, I'm actually from Ionis Pharmaceuticals, so I know Doug quite well. I just wanted to comment on the natural history thing because I think one of the things that we've seen in multiple disease areas, since we cover so many different areas, is that there's a lot of industry consortiums being created by the patient groups, and that's an opportunity to do your natural history study. So, because the patient group, I think, should own their natural history study. The patient should own their data for that, and everybody benefits. So I think that's another thing to think about for small groups, because there are multiple biotech companies and large pharma interested in all of these diseases right now, and so there's opportunities to get them to work together to help you. So I think I would encourage that.
Hi, my name is Alejandra, and I have a question for you. We are really, really small. We are just starting. Of course, we don't have none of the things that you guys have. You are huge. How do you create a patient portal or where you can start to register. We have the support of pharmaceutical, but they own their own database for the patients around the world. We have some patients in Australia, United Kingdom, etc. We have a support group that we support as much as we can. We actually help the doctors to find more information about the disease, but we don't own anything. So if you can help us to see what is the next step that we can, because I think that we own and we deserve to have something in our own.
So we had a few physicians in the beginning that championed the idea that there should be a single registry. And so they really, whether when they were talking to other institutions or pharmaceutical companies, when I mentioned the University of Pennsylvania, they really championed that notion that there should be a single registry and kept pointing people back to the patient organization. So that was something that helped us. We did build our own registry platform. We use a company called Digital Infusion, but I know that organizations like NORD have a great, I haven't used it, so is there anyone in the room that uses the NORD platform? There's some people back here that you could talk to afterwards, but they have a really, I think it's a pretty low cost way for you to build a patient registry through software and a system that they already have built. So if you're small and getting started, I would recommend that that's where you would go first. But you do need to find a champion in the community that's really going to keep pounding that idea of single registry. Because when you're a patient with a rare disease, you've already got 1,000 things going on. You're not going to put your information in this person's registry and then, I'm going to do it now for the patient organization too. That single registry is critical.
I think we have time for one more question. Does anybody else? One more. And just so you know, for those of you that have questions, if there's no time to get it answered, you can ask your question on the mobile app, and they will get answered.
Thank you. My question is for the second and third speaker. I noted that you wrote MTA on your slides, material transfer agreements, and I was curious to hear your perspectives on that process. The process of putting the material transfer agreement in place.
So someone owns everything. And so you need a legal document to transfer, let's say, to cell line, an antibody, a patient sample. Hopefully they're free and clear of other IP and you can give them out freely. So the slide that I think you're referring to is the example of our biorepository at Coriell, and I think it even said a simple MTA where you're just promising not to give it to a third party or to commercialize what you're getting. So it's you can use it freely for basic research. There are other more complicated MTAs depending upon the actual material, but we strive to make everything as freely accessible as possible. But you know, one lab can send another lab a mouse, but when it comes to a foundation and we want to protect our own interests, we really need a legal document in place when we're going to distribute things like that.
I don't think I have anything else to add. I mean, ours is a very similar situation. I think that the main thing I would say is kind of what I iterated a moment ago is that we treat this like a business, and that whatever legal protocols and procedures we need to have in place that an academic institution would have in place or a pharma or a biotech would, we do the same thing. So.
Okay, well, I think that'll end it there. That was a fantastic presentation. Thank you so much. Thank you, Hans. Thank you, Michelle. Thank you, Douglas, for that. For a great discussion and insights. At this time, we ask that you jump onto the mobile app and take our brief survey. And then enjoy the networking break coming up, which is sponsored by Spark Therapeutics. Our next breakout session will begin at 11:30. Thanks for attending the session and enjoy your week.